Science rules, and so do your genes. Have you ever wondered why you have your mother's eyes or your father's smile? Or maybe why some flowers are red while others are white? The answer, my friends, is all around us and inside of us. It's a fascinating field of science called genetics. Consider this: every living thing, from the tallest tree to the tiniest insect, has a set of instructions inside its cells. This instruction manual tells it how to grow, what to look like, and even how to act. It's the ultimate blueprint for life, and understanding it is one of the coolest adventures in all of science. It's what makes you uniquely you. Genetics is not just some complicated topic for scientists in white lab coats. It's happening inside you right now. It's the reason your hair is curly or straight, why you can roll your tongue or why you can't, and why you are a certain height. This amazing code of life is passed down from one generation to the next, a process we call heredity. It's like a family recipe book that gets passed from parents to children, with a few personal tweaks added along the way. So, how do we begin to read this incredible instruction manual? Before we can start mixing and matching traits, we need to know the language of genetics. Let's start with the most important word of all: gene. A gene is a small section of your DNA. Think of your DNA as a giant cookbook filled with recipes for making you. Each individual recipe is a gene. You have a gene for your hair color, a gene for your eye color, a gene for how tall you are, and so on. These genes carry the information that determines your traits. You get your genes from your biological parents, inheriting one set from each. Now, while a gene is like a recipe for a trait, say eye color, these different versions are called alleles. For the eye color gene, you might have an allele for brown eyes and an allele for blue eyes. So, the gene is the category, eye color, and the alleles are the options within that category, like brown, blue, green, and so on. For every gene, you get two alleles, one from each parent. This combination of alleles is what ultimately decides what trait you will show to the world. It's this pairing of alleles that makes genetics so interesting. Now that we know we get two alleles for every gene, one from each parent, a new question comes up. What happens if you get two different alleles? What if you get an allele for brown eyes from one parent and an allele for blue eyes from the other? Which one wins? This is where the concepts of dominant and recessive alleles come into play. A dominant allele is like the loudest person in the room. If it's there, it's the one you're going to notice. A dominant trait will show up even if only one copy is present. On the other side, we have the recessive allele. A recessive allele is a bit more shy, like a quiet whisper. A recessive trait appears only with two copies. If a dominant allele is present, it masks the recessive one. Brown eye allele is dominant over blue eye allele. One brown plus one blue allele. Brown eyes. To get blue eyes, you need the recessive blue allele from both parents. Two copies let the quiet whisper be heard. Scientists use letters to represent alleles, capital letter for dominant, B, brown, and lowercase for recessive, B, blue. See? The capital letter always wins if it's there. Alright, we've got our vocabulary down. We know about genes, alleles, and the difference between dominant and recessive. Now let's put it all together and talk about a hybrid cross. A hybrid cross is an experiment where we mate or cross two parents that are hybrids for a specific trait. A hybrid means the parent has two different alleles for a gene, one dominant, one recessive. Using our letter system, a hybrid parent would have the allele combination BB. They carry both dominant and recessive information. Why would scientists do this? Because it's a powerful tool to understand heredity. Crossing two hybrid parents shows all possible allele combinations in offspring. It lets hidden recessive traits reappear in the next generation. It's a way to test predictions and confirm inheritance rules. Think of shuffling two decks of cards and dealing all possible hands. The most basic hybrid cross is a monohybrid cross. Mono means one. It looks at one trait, for example, flower color or seed shape. For example, we might cross two pea plants hybrid for height. Both parents would be tall because tall is dominant, but both carry the recessive allele for short. So, what will their offspring look like, all tall or some short? A hybrid cross answers that with remarkable accuracy. So, how do we make these predictions about what the offspring will look like? We don't just have to guess. We can use a simple but brilliant tool called a Punnett square. A Punnett square is basically a chart that helps us visualize all the possible combinations of alleles offspring can inherit. It was invented by a scientist named Reginald Punnett. It makes genetics much, much easier to understand. Think of it as a genetic multiplication table. Let's walk through how to make one. Draw a square and divide it into 4 boxes. Know the alleles of the two parents. Our hybrid parents are B. Write B and B across the top, one per column. Write B and B down the side, one per row. You've set up your playing field. Now, filling the boxes. Combine the top letter with the side letter for each box. Top left, B plus B, BB. Top right, B plus B, B. Conventionally, we write the capital letter first. Bottom left, B plus B, B. Bottom right, B plus B, B. The 4 boxes show the possible offspring combinations. The combinations, BB, 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 BB. This square tells us probabilities. BB, 1 and 4. BB, 2 and 4 or 1 and 2. BB, 1 and 4. This little square is one of the most useful tools in genetics. We can't talk about hybrid crosses without talking about the person who figured it all out, Gregor Mendel. Mendel was a monk and a scientist who lived in the 1800s, and he is often called the father of modern genetics. He conducted thousands of experiments on a very simple organism, the common pea plant. He chose pea plants because they grow quickly, have many easily observable traits, and he could control which plants pollinated which. He was essentially performing very carefully planned genetic crosses long before we even knew what DNA was. Mendel studied 7 different traits: flower color, purple or white seed shape, round or wrinkled plant height, tall or short. Let's look at his experiments with flower color. He started with true breeding plants, meaning plants that always produced offspring with the same trait. So, his true breeding purple-flowered plants always made more purple-flowered plants. He crossed a true breeding purple-flowered plant with a true breeding white-flowered plant. What do you think he got? Every single one of the offspring had purple flowers. This told Mendel that the purple flower allele was dominant over the white flower allele. These first-generation offspring were hybrids. They had one allele for purple flowers, P, and one for white flowers, P. Their genotype was PEEP. But here comes the brilliant part. Mendel then performed a hybrid cross. He took 2 of these hybrid PEEP plants and crossed them. When Mendel counted the second generation, about 3/4 had purple flowers and 1/4 had white flowers. The white flower trait had reappeared. Mendel's principles don't just apply to pea plants, they're universal laws of heredity. They work in the animal kingdom, even in humans. Breeders have used these rules for centuries with dogs, cats, horses, and livestock. They were doing genetic crosses all along, breeding for stronger, faster animals with specific coat colors. Example: guinea pigs. Black fur allele equals B, which is dominant. White fur allele equals B, which is recessive. Cross a true breeding black BB with a true breeding white BB. All offspring will be hybrids BB. All the babies show black fur, but they carry the hidden recessive B allele. Now take 2 of those hybrid BB guinea pigs and mate them. What will their offspring look like? Let's set up a Punnett square with B and B across the top and down the side. Cell results: BB, 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 BB. What does this mean for fur color? BB and the 2 B guinea pigs will have black fur because B is dominant, but that BB combination inherits recessive B from both parents. As a result, it will have white fur, so even though both parents look black, they can produce a white pup. The predictable ratio is about 3 black to 1 white. The same logic applies to other animal traits like the polled trait in cattle and inherited health conditions in purebred dogs. The patterns are everywhere once you know what to look for. When we look at the results of a hybrid cross, like the one we did with the guinea pigs, we need 2 more scientific terms: genotype, phenotype. They sound similar, but mean very different things. The genotype is the organism's genetic makeup. It's the combination of alleles for a gene. In our cross, the offspring genotypes were BB, BB, BB. The genotype is written in the genetic code. The phenotype is the observable physical trait. It's what you see. Our guinea pig phenotypes: black fur, white fur. The phenotype is the result of the genotype. Different genotypes can give the same phenotype. BB produced black fur. BB produced black fur. That's due to the dominant allele. BB produced white fur, so you can't always tell genotype from phenotype. Let's analyze our BB by BB cross. Genotypic ratio =1, 2, 1. It shows distribution of allele combinations. Phenotypic ratio =3, 1. This 3, 1 is classic for a monohybrid cross and what Mendel found. So, we've explored genes, alleles, Punnett squares, and the classic results of a hybrid cross. You might be thinking, this is really neat, but what's the point? The point, my friends, is that this knowledge is incredibly powerful and has changed our world in profound ways. Understanding hybrid crosses and the principles of heredity is not just a classroom exercise, it is the foundation of modern agriculture and medicine. It allows us to make informed decisions that can improve lives and help feed a growing global population. Science is not just about knowing, it's about doing. Think about agriculture. For thousands of years, farmers have been trying to grow better crops, plants that produce more food, resist diseases, and survive droughts. Hybrid crosses give plant breeders a scientific method to do this. They can identify plants with desirable traits like high yield and pest resistance, and cross them to create new hybrid varieties that combine the best qualities of both parents. The corn you eat, the tomatoes you buy, and the wheat used for your bread are likely all products of sophisticated hybrid breeding programs. This process creates hybrid vigor, or heterosis. Often the hybrid offspring are stronger, healthier, and more productive than either parent. By carefully selecting parents and performing crosses, scientists create superplants tailored for specific environments. This led to the Green Revolution. It has helped us feed billions of people. It all goes back to the simple principles that Gregor Mendel discovered in his garden. Beyond the farm, understanding genetic crosses is vital in human health. Genetic counselors use these same principles to help families understand the risk of inheriting genetic diseases. They can predict the probability that a child might inherit conditions like cystic fibrosis or sickle cell anemia, which are caused by recessive alleles. This knowledge empowers people to make important decisions about their health and their families. From the food on our plates to the medicine that keeps us healthy, the legacy of the hybrid cross is all around us. And that is truly amazing.